All right, so now we are going to go over the second part of chapter one, starting off with the history of microbiology. So, I just picked some different things that I think are the turning points for microbiology over the past 200, 300 years. And again, I want to reiterate that microbiology didn't really kick off until the microscope was invented. And there was a guy in 1665, his name was Robert Hooke. He was kind of one of the main scientists of the day. He was kind of in a little bit of everything, physics, biology. Anyways, he was had a microscope and he was looking at all kinds of things, and he looked at cork. And he noticed the cork, which is dead plant material, was split up into little boxes. He called them cells. So that was the first time cells were discovered, and the name stuck. And around that time, there was a lot of people trying to improve on the microscope and make it better. And there was a guy named Antony van Leeuwenhoek, who was a Dutch merchant. Basically, he was a fabric merchant. So he was really good at, he had to make magnifying glasses to look at fabric to make sure it was high quality. And he was really good at that. He was really good at grinding lenses down. And he made himself a simple microscope with just one lens to magnify things. And he is the first person to see microbes. And you can see some of his drawings there. Um, Now, he didn't know what he was looking at. He called them wee animalcules. At the time, people thought that there were little dragons and stuff swimming around their teeth. He looked at teeth scraping, all kinds of nasty stuff. And he sent this stuff back to the Royal Society, and then people started to realize that there are these microbes that exist, and they were trying to figure out what they were. And one big thing that was going on at this time was that people believed life could generate spontaneously. So it's called the spontaneous generation theory. People thought that if you had all the ingredients for life, that things could magically spring to life. So think of it this way. If you were at a pond and you saw a frog just pop out of the water all of a sudden, some people would say, according to spontaneous generation theory, that frog just came into life right then, right there. Um, so this was a problem because this wasn't true. All living things come from other living things. And a lot of people were trying to prove that living things beget living things. You couldn't just have life pop into, there was no Harry Potter stuff, no popping in and out of existence. So the other problem that came along at this time was people thought all you had to have was a vital force. There was something in the air, they said, that was the vital principle or the vital force that gave the power to give life spontaneously. So if you guys look at your notes, there's a picture of the Francesco Redi experiment. There at the time there was another one. Somebody else who did an experiment that showed that if you boiled something, that you killed everything and life couldn't arise. But they said, you took away the air. So here's what Francesco Redi did. He was a doctor in Italy, and he said, listen, I got meat. I leave meat out, open up, I get maggots on it. People would say, if they believed in the spontaneous generation theory, that the maggots came from the meat, but actually they came from flies that came in and laid eggs on them. Then he had a sealed jar, and he let the meat rot. And lo and behold, there was no maggots because they couldn't get in. So the other scientists said, no, you can't do that because you cut off the air. And the air has a vital principle, which makes it impossible for life to arise if you don't have any vital principle. So then he took a mesh covering on the jar, and the flies still could not get in. I guess they must have laid their eggs on the mesh because the maggots showed up at the top. But you can see there's no maggots on the meat. So he showed that even if you allowed air in there, the maggots would not arise spontaneously. That was the flies that were laying the eggs. So people say, okay, you're right. But they wouldn't believe that for bacteria. At the time, there was a guy named Rudolf Virchow who was the Grand High Poobah when it came to biology in Germany. And he said, listen, all cells only arise from pre-existing cells. This is the start of what's called the cell theory, or biogenesis. And the problem was he had a hard time proving it in the lab. So in order to disprove spontaneous generation, you needed two things. You needed to show that if all microbes were killed beforehand, you had to have a sterile solution, that they wouldn't come back on their own. But you also had to expose it to air for the vital principle that didn't exist. Now, you guys should know that there is microbes in the air, and there are spores and all kinds of stuff in the air that if you expose something to the air, it's not sterile. So enter Louis Pasteur. It's 1861. He does his swan neck flask experiment. What he did is he boiled some beef broth. Beef broth is great for growing bacteria. And he put them in these long neck flasks. And then he bent, you guys, hopefully you can see this, he bent the neck into kind of like an S or shape. This is the swan necked flask. Now, what he did is when he boiled it, he killed the bacteria in there so they weren't coming back. This S shape allowed for air to go in, okay? But here's the thing, the microbes are heavy, so if they got blown in here, they would settle down here. They didn't have, mountain climbing equipment so they could climb into the beef broth. They get stuck down here. So he had a sterile solution, no microbes would grow. They couldn't say there was no air because the air could still get in via the neck, but it's just the bacteria would get stuck down here at the bottom and could not contaminate. And they still have one or two of these things today in museums in France that they still don't have any growth in them. And it's a lot later, it's 150 years later. So that killed. So next on, Louis Pasteur, who was like pretty much the very first microbiologist. He's the first guy that went pro, even though he was a chemist. He turned then to look at the germ theory disease. He was also trying to figure out fermentation and all kinds of stuff. But at the time, if you got sick, it was because you were bad or you talked in church or something like that. So people didn't know why they got sick. People were very scared. Bad things happened when there was outbreaks of disease. So Louis Pasteur wanted to show that germs, animalcules, bacteria cause disease. Okay? And he had a hard time proving that. And while he was working on it, he had some success, but a guy named Robert Koch, who was a doctor, a country doctor in Germany, came in and he actually proved that anthrax, which was something that killed cattle back then, farmers would get sick too, but mostly it was killed cattle. He proved that that was caused by a bacteria called Bacillus anthracis. And he also came up with rules to help prove that something is being caused by a microbe, and they're called Koch's postulates, and we still use them today to some extent. So first off, what he would do is he would take a sick animal that had anthrax, and he would make sure that he would find this bacteria, this bacillus, in every case, every animal that had the disease. What he would do is he would isolate it, and he'd grow it in a pure culture to where it was by itself. He started off, he did this in fluid of cow's eyes. He would get some of that aqueous humor and throw it in there. Later on, he moved on to plates and things like that. But anyways, he would take that bacteria. He started doing this on his daughter's pet mice. But he would inject them with the bacteria in the aqueous humor, and then they would die of the anthrax. And then the fourth step, he would isolate the bacillus again from the healthy organism. He would show, look, this bacteria, I put it in a healthy animal, it died, and it's still in there. So this is what's killing this is what's causing this particular disease. So it was a 4-step process. In fact, his daughter's mice were white. They were a special breed of mice. They were white. So the white mouse, the medical mouse that we usually see, that was the first example of that being. Oh, and here's a nice little picture. I'll just go through this with you guys. All right, I got a sick animal. I take microbes from that and I grow them in pure culture and I identify them with the microscope just to make sure we'll see what I got. Today we use fancier methods of identifying things, but all Koch had was a microscope. He'd then take from one of these colonies and he would inject it into one of his daughter's healthy white mice. If the mouse died, that means that's bad and that it had the disease. And then he would go in there and he'd culture more bacteria and make sure it was the same bacteria in the healthy donor that it was from the original sick animal. And then that was the causative relationship proven. Now he did this with 3 diseases, anthrax, with tuberculosis, and with cholera. I think he won the Nobel Prize for tuberculosis, discovering that. And yeah, it was tuberculosis. Now, there's some exceptions to that rule. So there are some bacteria. Get them in pure culture. You have to grow them on artificial media. He did not had some bacteria that you couldn't really do that with. So Treponema pallidum, which causes syphilis, is very fastidious. If you don't know what fastidious means, it means picky in this case. It doesn't like to grow on artificial media. Same thing with Mycobacterium leprae. So those were very hard to prove what that they cause. And then there are some diseases that there are no animal guinea pigs. There are only humans. So it would be not moral, not acceptable, not ethical to take HIV and infect somebody with it just to see if it would cause HIV, cause AIDS. So there are some exceptions. Let's take a look. Now, some other breakthroughs that kind of happened with Koch and his students, people that worked for him. They went from the aqueous humor, the cow eye, then they started using boiled potatoes to grow the bacteria to get pure cultures. Then they started using gelatin to make a semi-solid agent. The only problem with gelatin is that some bacteria can break it down, so they had to use something else. Walter Hetz was a worker for Koch whose wife used agar to make jelly, and agar is not able to be broken down by bacteria, so they started using that to make semi-solid plates to get columns. And Julius Petri also worked for him, and that's where we get the Petri dish. The shape of that was used to grow the bacteria. Okay, so here are some what we call the golden age of microbiology. Golden age of microbiology. You got Louis Pasteur. He comes in and he explains how fermentation works. You got. He also disproved spontaneous generation with his Sonnach blast theory. He came up with pasteurization. Before pasteurization, if you drank milk, you had a good chance of getting TB from it. So that was good that he came up with that. They'll call it today pasteurization after him. The other things he did, he came up with the rabies vaccine, all kinds of other things that this guy did a lot for microbiology. He generally feuded with Koch. They didn't get along, partly because one was French and one was German. If you guys know anything about Germany and France, from the 1800s to the early mid-1900s, they were at war with each other pretty much constantly. So they didn't get along. Joseph Lister was kind of their buddy in between them. He was an English surgeon who tried to get them to get along. His big contribution was he used phenol as the first antiseptic. What you guys got to realize is surgeons before this, they would sometimes just wipe their scalpel off on their boot and then go to cut. He said, listen, we got to be clean. We got to wash people off. We got to use antiseptics to try to prevent wounds. Koch came up with the postulates and kind of cemented the germ theory. He also kind of fell into disrepute because he tried to use tuberculin as a cure for TB, and it wasn't. The stuff that you guys, if you ever had a PPD test to see if you have tuberculosis, that stuff, he invented that back in 100. He thought it was a cure. It wasn't. People got sick, so he ended up not doing great at the end, but he's still remembered for his postulates. A guy named Hans Gram came up with gram staining, which is still used today in pretty much every hospital on the planet to help identify bacteria. He was looking for the bacteria that causes pneumonia. Paul Ehrlich, this is a guy who also worked for Koch. He was looking for drugs that would kill or attack bacteria and leave the body alone. So he was trying to find antibiotics, basically. He didn't quite do it. He came up with a synthetic drug called salvarsan. It was arsenic based, and it was meant to treat for syphilis. It didn't work that great, but technically it was the first drug against a bacteria. Outliers. Before all this happened, Edward Jenner came up with vaccination. He gave people cowpox to prevent them from getting smallpox. That vaccine is still working today. Most people don't get it anymore because smallpox has been eradicated. Vacca means cow, so you get cowpox. That's what vaccination comes from. After all this happened, Alexander Fleming discovered penicillin in 1928 on a plate that was growing Staph aureus. It accidentally got contaminated with mold. and the happy accident, he realized he could make antibiotics from that. It took another 15 years before they fully ramped up penicillin production, but he gets a lot of the credit for that. And then Rebecca Lansfield identifying stereotypes of streptococcus. So basically, she was looking at antibodies and how they react to different strains of streptococcus species. So she helped kind of help us realize that not only did we have all these different species of bacteria, but we had different strains that have different impacts on the body. There is if you look at your book has it split up into a first age, golden age, second golden age, the third golden age, which technically I guess we're in right now, where we're looking at all this bioengineering and GMOs, and we're also looking at the human microbiome. A lot of exciting things are happening in microbiology today, and hopefully we'll get a chance to talk about them. Okay, let's stop it there.